Ilya B. Silov. Ilya B. Silov is a Russian-American biochemist, molecular toxicologist, and virologist. He is known for his studies and research on enzymology, drug metabolism, environmental toxicology, bioinformatics, virology, and cancer. Silov has authored 4 monographs and co-authored over 280 publications on microsomal, purified and recombinant monooxygenases, and mechanisms of CYP induction by xenobiotic. Education and career. Silov graduated in 1970 from Novosibirsk State Medical Academy, USSR, and received a PhD degree in biochemistry from the same institution 1973. In 1983, Silov earned Higher Doctor of Sciences degree biochemistry from the Institute of Experimental Medicine, Russian Academy of Sciences. He held various positions at Institute of Molecular Biology and Biophysics in Russian Academy of Sciences, including head of laboratory and chair of the Department of Xenobiochemistry. In 1990, the U.S. National Institutes of Health NIH hired him for a position of a senior enzymologist at the Laboratory of Molecular Carcinogenesis headed by Harry Gelboin, National Cancer Institute. Since 1994, he held positions of research associate professor/professor/visiting professor at the Department of Medicine, Mount Sinai School of Medicine, Icahn School of Medicine at Mount Sinai, New York, NY. In 2005, Silov founded Xenotox, Inc., a life sciences consulting company implementing an earlier He was elected an emeritus member of International Society for Study of Xenobiotics ISCS. Dr. Silov has lectured and keynoted at research seminars and conferences on many occasions. Research. Silov used protinophores and probes affecting arachidonic acid of phospholipids in endoplasmic reticulum to show different compartmentalized hydrophobic clusters coupled with substrate binding and electron accept. Following up discovery of mechanism for phenobarbital type induction of CYP2B6 and applicability of induced fit hypothesis for membrane-bound enzymes, Silov with Konstantin Gerasimov prioritized method how to convert first-line antipyretic and antimalarial medicines into their long-acting forms. Silov and Dmitry Oshchapov revealed numerous DRE genes encoding XAP2 and HSP98A on chaperone proteins complexing with ligand-free cytoplasmic A receptor. Silov also conducted research on cDNA-expressed mammalian CYP as preferred tests for major medical conditions. Silov revealed that human CYP1 is the catalyst of ethanol and uroporphyrinogen oxidation. Silov and Andrei Pokrovsky discovered TCDD ability to increase HIV-1 reverse transcriptase activity, HIV-1p24 antigen, and augment HIV-1 LTR-CAT construct in HIV-1 infected CD4 plus cells, all mediated via the R/B. Selected publications: Silov, I.B. Gromova, O.A. Lyakovich V.V., October 1976. Mechanism of inhibition by carbonyl cyanide in chlorophenylhydrazone and sodium deoxycholate of cytochrome P-450 catalyzed hepatic microsomal drug metabolism. The Biochemical Journal, 16175-83. doi 10.1042/Bion 600,075. I.B. Zakharova Polikova, N.E. Gromova, O.A. Pospelova, L.N. Lyakovich, V.V. October 1978. An in vivo in vitro comparison of the effects of bile acids on the structural organization and functional activity of liver microsomal monooxygenases. Experimental and Molecular Pathology. 20-131-143. doi 10.1016/14-4000, 878-90033-3. Silov, I.B. Zakharova, N.E. Gromova, O.A. Lyakovich, V.V. January 1976. possible mechanism of induction of liver microsomal monooxygenase by phenobarbital. Biochimica at Biophysica Acta 421-44-56. doi 10.1016/300-4-165-76-90,168-9. Silov, I.B. Gerasimov, K.G. May 1986. New approach to production of prolonged-acting medicines, facilitating hepatic drug detoxification system. Patent SU/EP 1,457,593 Aeon Ford G1 and 33/48. Nedoskina, E.A. Ostyapov, D.U. Mordvinov, V.A. Katakin, A.V. Kuznetsova, T.N. Shamanina M. Silov, I.B. August 2007. Detection of new potentially active DRE sites in the regulatory region of human genes encoding components of our receptor cytosolic complex. Organohalogen compounds 6912, 1890-1894. ISIN 1026-4892. Silov, I.B. Goldfarb I.S. Gelboin, H.V. December 1993. Enzyme kinetic and immunochemical characteristics of mouse cDNA expressed, microsomal, and purified CY-PONAON and CY-PONATWO. Archives of Biochemistry and Biophysics 307, 259-266. doi 10.1006/Abby 1993.1588. Silov, I.B. Mikhailko, V.M. Gelboin, H.V. March 1994. Isozyme and species-specific susceptibility of cDNA-expressed CY-PONA P-450s to different flavonoids. Biochimica at Biophysica Acta 1205, 325-335. doi 10.1016/167-4000-838-94-90,000, 252-6. Silov, I.B. Pokrovsky, A.G. September 1993. Stimulatory effect of the CY-PONAON inducer 2378 tetrachlorodiazo-P-dioxin on the reproduction of HIV-1 in human lymphoid cell culture. Xenobiotica 23, 457-467. doi 10.3109/498 trillion, 259,309,057,034. Silov, I.B. Scher, I.N. August 2008. Mechanistic insight into activation by dioxin-type xenobiotics of cancer-linked common human viruses and HIV-1 virus. The Lancet Infectious Diseases 8/496-9. doi 10.1016/70172-5. Silov, I.B. Scher, I.N. Ostyapov, D.U. Pokrovsky, A.G. June 2012. Xenobiotical Virology, Novel Mechanistic Concept of Hazardous Human Viruses Upregulation by Body Burden Level of Dioxins via AR-Mediated Transcriptional Pathway. International Journal of Infectious Diseases 16 Supple. 1. Eon 112-9. doi.org/10.1016/J. Igit. 2012.5, 258. Eon 112,